thank you very much for the invitation, and thank you for coming. So I'll be talking about work that I've been doing over the last couple of years with a lot of people here on generally trying to understand the immune system. So I'll explain what I mean by that. So it's been work mainly in collaboration with Thierry Morat. And the first part of the work that I'll talk about is with Yuval El-Hanati and Quentin Marcoux and Kurt Callan. And then I'll mention some work done by Andreas Meyer with Vijay Balasubramanian and us. And finally, if I have time, I'll hopefully tell you about some new results we have with our Russian friends, which has been bravely led by Misha Pagarelli. Okay. So, but first I'd like to take a step back, since this is a general talk and a lot of you are sort of thinking, oh, all this biophysics coming up here and now. I'd like to make a point that biophysics isn't anything new, and if we think about quantitative approaches to biological systems, this takes us back at least 400 years, if not way back, to the experiments of Robert Hooke, who observed cells under the microscope, living cells in a quantitative way. And if you look at this, well, once you stop thinking it's just a blob, you actually see that these are cells. And so this tradition of quantitative biophysics has continued with, of course, the observation of pollen moving in fluids, which so Brownian motion, which then led Einstein to develop his groundbreaking theories. New microscopy with Ramon y Cajal observing nerve cells in a very dense medium. The groundbreaking Luria-Delbrück experiment, which is really an amazing experiment based on the idea that exploits stochastic processes to show us that mutations arise randomly and are not stimulated by external stimuli. The first attempts at sort of what we now call mathematical modeling of description of action potentials in neurons. This curly-whirly piece of polymer, which you all recognize, the double helix, and the limits to sensing by cells of gradients and molecules, the basically precision of cell sensing. So as you can see, there's a long tradition of trying to understand systems that are living and seem quite complex. The other thing that I'd sort of like to say is that in biology, there's a lot of basically everybody predicts. This is a neutrophil, so a part of your adaptive immune system going after a bacteria, and it's chasing it, and it has to figure out where to go. It has to figure out where the gradient will go. These are E. coli swimming in looking for food sources, and they also have to figure out in a noisy environment where food is. And this is my favorite, newest example, which I learned this summer from Antonio Celani, which is the moth. This is a male moth, and the male moth is born, hatched from a larva without a digestive system. And it has a short amount of time, a few hours, to find the female moth and reproduce. But of course, a moth lives in a turbulent environment of this air. The female emits pheromones to give signals to the male, and the male has as much food as it's stocked up, being a larva, to find the female. And if it fails, it dies and doesn't manage to reproduce. So it really has to make predictive decisions to fly in the right direction. Otherwise, its life is completely pointless. Isn't that a beautiful system? So another system that has to predict the future all the time is the immune system. So I'll be talking about our adaptive immune system. I'll tell you a bit more about it. And the basic aim of the game is that there's pathogens out there all the time. They're trying to attack us. And we have an immune system which recognizes these pathogens and protects us. However, if you know, if your immune system is just chasing after the current pathogens, you'd constantly be sick, you'd constantly be, you know, at least under the weather and not feeling well. So there has to be some element of prediction in it. And I'll tell you how a lot of diversity, well, basically how the immune system manages to do it. Partly through our own work, partly through known things. So how does it work? Well, the immune system consists of cells that are called B and T cells, the adaptive immune system. And these cells express receptors on the surfaces, and these receptors recognize foreign pathogens. So it's a receptor just as any receptor, like a photon receptor, only it's a specialized receptor that can recognize only some types of molecules. So if it recognizes a pathogen, then it triggers an immune response. However, one type of cell has many receptors on its surface, but it has only one type of receptor. So it can only recognize a small part, not all the possible pathogens out there, but only a small part of it. And since there's many different pathogens in the world, we need many different receptors to protect ourselves against them. That's the basic idea. So, okay, one cell, one type of receptor. One type of receptor, you can think in the simplest way, can only recognize one pathogen. That's not true. It can recognize a few, but a small fraction. So since you have a large space, you can think of it as essentially, well, a very large space, then you need many different types of receptors to protect you against the many different threats. So the question that I'm interested in is, well, essentially, how does it work? And to phrase it in a more concrete way, what is the diversity of these immune receptors that allows us to protect us against these pathogens? And by us, I mean normal people, or normal individuals. So anybody who's essentially healthy, gets sick from time to time, but doesn't have any significant autoimmune disorders, as, you know, nothing serious, so that, you know, normal. And by natural, I also, for humans, it's not a problem, of course. For species and labs that are engineered, that's what I mean by natural. So to break up this question, I'll tell you a bit about how diverse is the immune system to be able to perform this task of recognizing pathogens. And then I'll take a step back and ask a more abstract question of, if I could design the optimal immune system, how would I do that? Okay. So first, just to give you an idea of why is it a problem? I mean, I sort of gave you an idea in terms of waving my hands. But as humans, we have of the order of 10 to the 8 unique T cell receptors, so unique cells. We also have of the order of 10 to the 4 genes. So if we wanted, so as you know, a receptor is a protein. Proteins are coded by genes. So if we wanted to code each receptor by a single gene, that would mean we would have to have 10 to the 4th more genes than we actually have. To give you an idea, so genes are in a part of your cell which is called the nucleus, and this basically wouldn't fit. So we have a volume problem. Then there's a sort of cuter representation of the volume problem, which is here on the example of a mouse. So a mouse, as you probably know, is the hero of immunology labs. Many mice have died in sacrifice to this science. So they have, and that is because they have an immune system very much like our own. And this is just a funny picture showing you the point that if it was one receptor against one pathogen, the immune system of a mouse would take 70 times the volume of the mouse. So again, that's not what's happening. There is some cross-reactivity. A receptor can recognize a few pathogens, and a pathogen can be recognized by a few receptors. But you see, we're starting to get into the problems of it has to be diverse, but we have a finite number of resources and even space, which normally in biology isn't such a big problem, to account for it. So nature has come up with a completely mind-blowing solution to this. And it relies on two notions of combinatorics and randomness. And this is how it works. So this is the cell with the receptor sticking out of it. There's a part which basically makes it stick into the cell, a constant part. And then there's the variable parts which do the recognition. Each of the variable parts consists of three parts called V, D, and J. They're just names. And the way they're put together is that in your genome, so as you know, each cell in your body has exactly the same DNA. So this is true. Each cell in your body has a number of genomic templates. It has of the order of 48 templates for V, 2 for D, and 13 for J. And in a combinatoric process, the cell picks one V, one D, and one J. So for example, here V9, D2, and J3. But you can see there's of the order of 1,000 possible combinatoric combinations of picking and choosing one of these segments and putting them together. Okay, so we've gotten to 1,000. But I told you we have of the order of 10 to the 8th unique receptors. So there's some more work to do here, a few orders of magnitude, a few serious orders of magnitude. So this isn't the end of the story. Then comes another set of enzymes, and they randomly, at the junctions between these segments, the V and the D and the D and the J, they randomly insert and delete nucleotides. Okay, this happens at the level of the DNA. The DNA is made out of nucleotides. So imagine you have a string. Sorry, my lifesaver is getting in my way. And then you just randomly take some out and put some in. Some letters in and some letters out. So you're basically scrambling these junctions. And this adds an additional level of stochasticity. So you can imagine doing this, you've done this, and since you've just scrambled things together, you can imagine that it produces something that completely doesn't work. When DNA is made into proteins, there's a code for doing this, and it has to happen in multiples of 3. Okay? And sometimes so basically 3 nucleotides make one amino acid, one part of the protein, but sometimes you have a 3-letter code that tells the system to stop. And you can easily imagine, since you're basically scrambling these things, you'll end up with something that will tell you to stop too often, or will not be in the right frame to make a protein. So this indeed does happen, but we have 2 chromosomes, and any animal that has an immune system, adaptive immune system, has 2 chromosomes. So you try with the first one. If it works, great, that's your receptor. If it doesn't work, you silence it, and you try with the second one. If the second one doesn't work, then the cell has to die. But if it does work, then you express the second one, and you keep the first one as a fossil record of this initial recombination process. The second one is functional, selected. Everything happens to the second one. You never see the first one, but it's in the cell. And we'll use this information in a sec. OK, so we just generated our receptors. And then the first thing that happens to them is they get tested for. They get asked the question, basically, of will you be a good receptor? This happens in the thymus, which is a part of your body which you sometimes see when it comes from the cow, and it's called sweetbread on your plate. But the interesting thing about the thymus is that it's a pure organ, meaning it only has molecules, proteins that are natural to your own body. It doesn't have any viruses, anything foreign, at least, if all is going well. So the test happens there in this clean environment. You can think of it like as a clean room where you're testing things. And the cells get asked 2 questions. First of all, will you bind any protein at all? If you bind nothing at all, then you're probably not a very good receptor protein. That's like building a receptor that never catches anything. So let's throw that away. However, if you bind too strongly to your own self-proteins, so you bind too strongly to things, you only have your own proteins in the system, then that's also not a good thing. So you're going to throw this cell out. So you keep the ones in the middle, and a group of cells made like this then goes out into the world and has the goal of recognizing foreign pathogens. If they recognize, they proliferate. Otherwise, they die. And at the end of this, we get what we call the repertoire. We get the set, the ensembles of cells with the receptors. And if we look at this, at the basically density of states of this distribution, so here it's plotted as the probability versus clone size rank, but it's exactly the same thing as the density of states reversed. So we basically take the most common receptor, give it rank 1, second most common rank 2, and then plot the appropriate probabilities. It's also the cumulative distribution function of the probability distribution of seeing a given receptor. The main thing you see about here is that it has very long tails. It's actually, it also has exponent 1, and we now understand why it is. But all I want to say at this point, this is data from a zebrafish, which also has an immune system like our own, is that it's very diverse. You're equally likely to find a clone that is very large as a clone that is very small, just because of the entropic contribution of the very small clones. So diversity is a fact. So this process, as I said, it's quite amazing, really. It does generate very large diversity. Okay, but if we want to really quantify how diverse is it, we first need to say something more quantitatively about how this initial diversity is generated. So far, I sort of told you the textbook picture. And this is where recent experiments come in. So as you may have heard, generally in biological science, there's been a sequencing revolution. And now we can sequence everything. And when we can sequence everything, we know everything. This isn't exactly true, because we still don't know much. But sequencing is a very powerful technique that allows us to go into the DNA or mRNA and basically read it out as we would read out a book. The only problem is that sometimes this book is in a language we completely don't understand. So, but you can do that with the immune system genes also. So with the receptors that have actually recombined. So what you do is you take blood from people, then you sort out the cells, you sort out the receptors, and you sequence them. And the goal of this is to, if you get good sampling, you basically have a list of all the immune receptors that are there in somebody's bloodstream. And you can do this for different people. You can compare them. So this is an experiment which was done in Hull and Robbins' group in Seattle, which took 9 people and looked at human T cells. These are the details of it, and their interest as well as ours is in this variable region where I told you was this stochastic chopping up and insertions and deletions of novelty, which the immunologists called CDR3, but it's basically this most random part. And as I told you before, I told you there's these 2 chromosomes, one that, remember that I said that the first one maybe didn't work, and then the second one got expressed. So since the only thing that happened to the first one that didn't work is that it got made. It never had any function. It never got tested. It never underwent any of those other steps. These are what we call out-of-frame reads. We can identify them that way, and that gives us insight into the generation process. Then we can look at the in-frame reads, which are the functional reads, and they give us insight into the selection process, what later happens to the actually functional ones. So basically from the ones that nothing ever happened to them but were made, we're going to build a baseline for which we can then characterize selection and how surprised we should be how things happen. Yes?
Why it's 14%? I mean, you should have more or less half of the cells that have the first one expressed and the other half that have...
No. So it's more complex. So it's a 3-letter alphabet, right? To be out of frame, it's a 1 chance in 3 of basically missing your thing. But because it's not, why isn't it 2/3 versus 1/3? Because there's a recovery rate. You can correct your mistakes, which I didn't want to tell you about, but nobody knows what this recovery rate is. So this is an experimental fact, which we've done these back of the envelope calculations, and we basically we can learn the recovery rate from knowing this is the answer, and it doesn't sound crazy, but it's not. Okay, but so you get the idea why you have these. So let's just focus on the ones that nothing ever happened to them but were generated. Okay, so as just a reminder of this process, combinatorics and randomness, and this results in these receptor sequences, which I said are just strings. And the goal of the game is, can we come up with a probability distribution that describes the probability of generating a given receptor, right? So that then you give me a sequence and I tell you whether this is likely to be a T cell that has been generated or not. I'll have this power. Okay, so the first thing we can do is the basic approach. Is we take a sequence, and for each sequence, we find the most probable scenario that produced this sequence. We match our sequence with the genomic templates, which we also have, and say, well, this sequence was probably produced by this V, this D, and this J, and then it had this number of insertions and deletions, and from this, we calculate the statistics of using a given V in the generation process, a given DJ, having a given number of deletions, and so on. But there's an obvious problem with this approach, which is illustrated here. So we have these two gray sequences, and they're exactly the same. Okay, you can now check that, but trust me, they're the same. And the point is that any sequence, because of this randomness, because of these insertions and deletions, you can generate in many different ways. So you could take this V2 and D1 and delete bits of them and add whatever you need to. Or you could take a V9 and a D2 and delete whatever you need to, and you get the idea, right? And you can go on and on and on. So basically what this tells us is that it's impossible to deterministically say how a given sequence was made. We need to do it probabilistically. So that's what we do. So for each sequence, instead of picking our favorite candidate for how it was made, we build a whole library of candidates. So on the previous slide, I show you 2, but you can easily imagine that there's many more. And then we assign probabilities or weights to each of the scenarios. From then, we calculate the probability of using a given V, and so on, and then numbers of deletions, insertions, and so on. And then we have a model from which we recalculate the probabilities of using them. So we do this in a probabilistic way. We sum over all possibilities, weighting all the possibilities. And we have this model, and basically this model is the most factorized, self-consistent model that works here. So we learn the weights and the elements of this model in a self-consistent way. And we want it to be the model to be as general as possible and as self-consistent as possible. Once we have that, we have the probability of each of these scenarios, the weights, and then we sum over all the scenarios that could have generated a given sequence to get the probability of generating a given sequence. Yeah?
Is it clear that the independence that you assume here, the statistical independent. Is that verified?
Yes. So we do it self-consistently. So for example, just to give you an idea, as I said, we wanted the most factorized form. So the first thing we assumed is that that PDJ factorizes into PD times PJ. We did that, and then when we looked at the correlations, we saw correlations between D and J. So we said, aha, that's a wrong model. We have to have this be adjoint because there is a dependence. Since we're physicists, that's how we did it. Then we went and looked at the textbooks, and there's a very good mechanistic reason for why this is correlated. Why basically the way it's on the genome architecture imposes this to be adjoint. And but yes, we basically verify it a posteriori and see that this form holds up. Okay, so this is where I'm going to connect to sort of current goings on. So we learned this model, the probabilistic model, and of course I won't go through all the elements of it because you probably don't care, but I'll just show you the probability of insertions. The blue line, solid blue line is for VD, and the solid red line is for DJ. And the first thing to notice is that there's also these dashed lines, which are the estimates you would get from the deterministic assignment. So, you know, in this day and age where we're wondering whether we have a new boson that's just been observed at CERN or whether it's just a fluke, this is a fluke. Okay, and when we first saw it, we were very excited. We had new biology here. We, reinvented the whole VDJ recombination process. But it's a statistical fluke. As reproducible as it is, your method's wrong. You reproducibly get the wrong thing. I sincerely hope for our colleagues at CERN that they have more luck. But this is the same kind of problem, right? The other fascinating thing here is that there's error bars here on this, and the error bars are from 9 individuals. 9 completely unrelated individuals, probably lab members in Seattle, so different ages, different ethnicities. And you see that, I mean, for biology, this error bar is essentially 0, right? The process is incredibly reproducible. So it's a very universal mechanism. So it doesn't matter, who your grandparents were, what you eat, or how old you are. And basically, the way you and I generate your receptors is exactly the same. So, yeah. Okay. So once they're generated, I told you that there's this step of checking whether they'll be good receptors. So they need to be selected upon. And so basically, then we need to describe this selection process. So that means that you generate a distribution like this, and in the selection process, some of them will get killed and never appear again. So again, just because we see a high fraction of some receptors here doesn't mean that they've been selected upon, because maybe it's very easy to generate this receptor. So we need a baseline. This is why we put all this effort into learning this initial generation problem, which is a baseline, which tells us how surprised should we be if we see a given receptor after the generation process. And I haven't dwelled on this too much, but in fact, this is a very ununiform distribution. So there's a very large basically it's a very heterogeneous process, which the generation probability varies over large orders of magnitude. I'll show you that in a second. So we need to correct for this, and to do that, we need to build a selection model. So we say that selection independently acts on the and J gene, and then on the length of this variable region, and then independently on each amino acid in the middle. We don't worry about the D gene because it's so short that it's very hard to say something about it. Again, there's a technical problem. It's not easy to characterize this post-selection distribution because it's a very large distribution with many, many rare events. So we can't learn this directly, but having a model allows us to learn this in a self-consistent manner using similar techniques as for the generation process. But I won't go into this in detail. I just want to show you the result about what we actually learned from this. So what's plotted here is the histogram of generation probabilities. First, focus on the blue line. That's the one I just described, the pre-selection histogram. So I take the generated repertoire, and I ask how many sequences have a probability of 10 to the minus 20 to be generated, how many 10 to the minus 10, and so on. Okay? Just the histogram for the distribution. So this is the generated, and the red line is the selected one. So as I said, the first thing to notice is that this goes from minus 5 to 10 to the 5 to 10 to the minus 25, so it's very non-uniform. But you also see that after selection, there's a shift to seeing sequences that were more likely to be generated. The post-selection probabilities have a higher probability to be generated. So what that means is that sequences that are more likely to be generated are over here, are more likely to be selected. So what that means is that the whole machinery that makes sequences is more likely to make sequences that are then more likely to be selected. So from the ground view of evolution, sequences, basically, this whole machinery has been shaped by natural selection, the real evolution, right? The thing that Darwin-like. And so that has evolved over the years to anticipate then what the little evolution in your body will do. So there's a matching of these two processes.
You say that there are cases when both variations don't work and the cell just dies.
Yeah.
And so in this case, you never observe.
We don't see them.
So there is some things that are generated, but you actually never see in your.
Yes. Yeah. No. So they. Okay. But to test, basically, to test for the bias that this could have imposed, what we did is once we have the model, we generate sequences, and then we see the same fraction of in frame and out of frame as we do in real life. So we will never, we will never see a cell that has to die because we never group them into cells. Then we could group, we assume that each of these generation events in the cell happens independently. So, but then we could take all the sequences we generated and then stochastically group them into cells, and a certain fraction of them would have double bads, and those would be the ones that die. But of course, we don't know what that number is. Since they die, we never see them. But we can see that when we generate de novo in silico, then we do see the right fraction of in frame and out of frame. So that's the test for what you're worried about. Okay. So to say something about the diversity, so to characterize the diversity, that's what we were initially interested in. We looked at the entropy of the repertoire, right? So I don't have to convince you that entropy is a measure of diversity. And so for the generated repertoire, if we look at now the only in-frame, so the good sequences, we get a generation entropy of 43.5 bits, which corresponds to a repertoire size, if all sequences were equally likely, which they're not, of an order of 10 to the 13. So I told you that we have of the order of 10 to the 8th unique, 10 to the 11th total T cells. This is much larger. So what that tells you is that the potential we have for generating diversity is much larger than we actually use, which is good, right? Because this is the pool from which we choose from, and since we're all choosing from the same pool, that's what I showed you with universality, you choose some part of random sample of this pool, maybe not random, and I choose another part of this pool, so that's how we all end up different with this large set of possibilities. The other thing to notice, and we have also numbers on this that I didn't put here, this is the entropy attributed to V and J, basically VDJ choice, the gene choice. This is the entropy attributed to insertions and deletions. So most of the diversity comes from insertions and deletions. And this is very much against what any immunologist will tell you, because when they classify these receptors, they just say it's this V, this D, and this gene. And we've told them that this is the wrong thing to do anyway, because there's no way they can actually do this. And second of all, they're missing essentially all of the diversity. So the next thing that happens is selection, and selection significantly reduces that diversity, gets us down to 38 bits. Again, most of the reduction is in insertions and deletions, which now corresponds to a repertoire size of 10 to the 11th sequences. And it corresponds, so going this basically 10 to the second reduction corresponds to a 50-fold reduction in diversity. Okay? So what should we be, I mean, it's a 50-fold reduction seems a lot, but it seems even more if I tell you that this process of selection, in terms of the numbers of sequences, it keeps 15% of sequences. For 15% of sequences, but only 2% of diversity. So again, we're saying the same thing we saw before, that it gets rid of the rare clones, and it favors, selection favors the clones that were likely to be generated. So we reduce diversity much more than we reduce the numbers. Okay, and last thing about data is getting back to the question of are we special? Because, you know, we all think we are so special, but if we look at how many receptors you and I share by chance, this is, I'm plotting basically the data versus a random expectation. Now that we've characterized the probability distribution, we have a random expectation. It's just p squared, right? Basically the probability of generating and selecting a sequence squared. And you see that the random expectations corresponds completely to what we observe by pure chance. So even at the level of our first functional repertoire, at the level of unique sequences, not taking into account, again, there's no difference between, well, there's no difference between any of the 9 people in Seattle. We can tell you there's no difference between people in Seattle and people in Moscow. So I'll say there's no difference between you and I also. Okay. So then the question we asked ourselves is, can we try and understand that in a bit more detail? And we took a step back and asked, what would an optimal distribution look like? So again, you have a set of pathogens, and should your optimal distribution be something like this, or maybe have more of these brown ones and less of something else? So there's a trade-off here. So, why can't you just track every pathogen in the world? Well, there's, I showed you the space problem, which we can formulate as a problem of having a limited number of encounters. If you have a distribution of receptors and a distribution of antigens, to recognize each other, they have to meet. But It's like meeting people by randomly in a big city. It doesn't happen every day that you meet everybody. It takes time. Physically, they have to meet. So the question we asked is, how should immune receptors be distributed? And I don't mean in space. I mean in, in terms of how many receptors you have specific to this pathogen as opposed to that, to minimize the harm from infections, right? Because presumably that's what the immune system does. It minimizes the harm from infections. So to make this a bit more precise, I'm going to have a receptor distribution PR and an antigen distribution QA. Antigen is just a bit of pathogen, same thing as pathogen, essentially. And I'm going to say that they have some recognition probability when they meet. So this is related to the fact that I told you, remember the mouse and the 70 times the mouse box? So there's this element called cross-reactivity that basically there's not a one-to-one mapping between receptors and antigen. So I'm going to assume that both receptors and antigen live in what I'll call recognition space or shape space. And it's basically the idea that if they're close together in this space, so like the blue ones here, the blue receptor versus the blue antigen, they have a high probability of recognition. And if they're far, then they have a slow probability of recognition. So the blue one is useless against the red one. But then there's the purple one, which, although weakly, can be recognized by both the red and the blue. Okay, so that's the basic idea. So to calculate the probability of recognizing any antigen, you have to sum over the probability that it's recognized by any of the receptors, and then with different rates by different receptors. Okay. So then we're going to measure our time in the mean number of encounters, and we're going to make the assumption that the harm caused by a given antigen increases with time. So an antigen appears, and your immune system tries to recognize it. As it doesn't, you get more and more sick. It eats away at your tissues. So finally it does, and things get better. So basically this idea is formulated here in terms of equations. Knowing the probability of recognizing a given antigen, we're going to say that the encounters are Poisson distributed in this the number of encounters. Sorry, the recognition events are Poisson distributed, and that the cost increases in some way, non-linear way possibly, with the number of encounters. So then to calculate the total cost from one antigen, we just have to integrate over time when the possible encounters could have taken place. And then there's a virulence prefactor, which basically says whether you're trying to recognize the flu or Ebola, you may want to act with more urgency in some cases than with others. Okay, and then this is against one antigen. So then you have to sum over the distribution of all antigens from the cost coming from all antigens. And the question we're asking is, what is the optimal repertoire? So can we optimize this cost function, assuming a fixed distribution of antigens? So for simplicity, for now, we're going to say that on the time scale on which we're trying to solve the problem, the antigen distribution does not change. So just to give, sum up, if we have an antigen distribution, which is the blue line, again, same blue line here and there, should we put our receptors only against the most common antigens? Should we distribute them a bit more uniformly and also take into some of the rarer ones? Okay, so this is the answer now. And I'm going to, for simplicity, consider the linear cost, but we considered many, many different costs. I just don't want to go into it. This corresponds to an exponentially expanding antigen population. So antigens expand exponentially in time. That's what they do. Viruses, bacteria, they grow exponentially in time. And the cost to your organism also grows exponentially in time. And if you do that, then this is the same blue line in this antigen distribution and recognition space that I just showed you on the cartoon. it's exactly the same. And the green line is the answer. It's the optimal repertoire distribution that we find from doing this optimization problem. And what you see is that it doesn't, you don't have the peaks where you have the most common antigens. It doesn't follow the antigen distribution, but it tiles space in a uniform, rather uniform manner. In such a way that if you look at the coverage against the antigens, that is fairly flat, and that does follow the antigen distribution, more or less. So you can understand this. So I told you about cross-reactivity. By placing a receptor here, think of a little Gaussian around there. That'll take care of some of the people, well, some of the antigens here. The other element, though, is what is important in the solution is that you actually have the element of protecting yourself not just against the common ones, but also against the rare ones. I don't have time to go into the details of the calculation, but we can explore this in more details analytically and show that the particularity of the solution is that it leaves proportionally more protection against the rare antigens than against the common ones. The idea one is that even if something's very rare, if you don't take care of it, it'll kill you, right? A rare event can kill you, or at least harm you. We don't have death explicitly. So those are the two elements of the solution. This is the same optimal solution now in 2D, in a two-dimensional recognition space, and you see that this speckle pattern gets reproduced. So the key elements are that cross-reactivity takes care of things, but on the other hand, you do want to protect yourself against the rare antigens, so you need to cover all of space. So we can analyze this by looking at the radial distribution function. And what this tells us is that there's a hard core, and then there's a distance at which we can place our receptors, a characteristic distance, so that receptors cannot be too close to each other, but they're as close to each other as possible, but remain excluding. So in terms of the structure factor, we see that at long wavelengths, so short distances, the precise local position is random. It doesn't matter. Cross-reactivity will take care of things, right? If you put a receptor roughly in the right region, it'll protect everybody around it. But at small wave vectors, so at large distances, everything is reproducible. So you have basically a reproducible picture. So this is what I've shown here with this cartoon. If you look at it from afar, it's exactly the same thing. But I've drawn the lines to guide your eyes to see that the receptors are actually not placed at all at the same positions, but you can't tell that from afar. So the question is, what does this mean for you and me? And so two individuals living in a given environment see the environment in a slightly different way, right? Just because, again, we're sampling, the environment is the same, but we're sampling it a bit differently. So say you're blue and I'm yellow. So the question is, yeah?
Sorry to interrupt, but how is it that there is an optimum distribution independent of the environment?
No, it's not independent of the environment. It's for a given fixed environment.
Oh.
So yeah, so we're in this environment, but we're both seeing a slightly different. So now I optimize mine from the one I'm sampling and yours for the one you're sampling. The question is, should we have the same optimal receptor distribution? The answer was on the previous slide, and is no. Because the precise positioning doesn't matter. So the similarity between our distributions as a function of how differently we see the environment very quickly goes to 0, even though the environments we're seeing are still essentially the same. They're slightly different, but still, essentially the same. And both of these distributions give us perfect coverage against their environment. So the bottom line is that we have very different repertoires, and we shouldn't be surprised by that. So the fact that we have a small overlap is basically shouldn't really surprise us. Of course, this is in recognition space, the overlap is in sequence space, but seeing that we have such a large dimensional space and such a complicated problem, we can all find equally good solutions to the same problem that are completely different. So this is just one last thing is just to say that the kind of solutions we get are completely consistent with traditional dynamics that has been considered for these kinds of systems. But I won't go into that. I want to end with the question of, are we all really the same? And so we looked at more individuals, and this is data coming from Dmitry Chudakov and Yuri Lebediev and Ilgar Mamedov's lab in Moscow. And so this is, again, the question of sharing. How many receptors do any two individuals share? This is what I showed you before. Well, not the same data, but the same idea, that for non-related individuals, for non-twins, even brothers and sisters and brothers and brothers and sisters and mothers and daughters and all that, it's all random. But then we look at identical twins, and they disagree. Identical twins are special. And if we look at, again, the histogram of the generation probability of the sequences that people share, this is just the one I showed you before. The red lines are sequences that are shared by non-related individuals. The blue is the theoretical expectation. The green line is the sequences shared by twins. And you clearly see an arm here, a long tail, and a decrease here. You see that the sequences shared by twins are somehow different in the generation probability than the sequences shared by everybody else. So what's so special about twins? Well, we know what's so special about twins. Identical twins are the only two people in the world who ever shed blood without a transfusion or anything additional like that. So they basically, in utero, the blood goes between one twin and the other, and they share B cells and T cells. So the mother and the child do not. There's a barrier in the placenta. They share blood, but they don't share. They don't actually share immune cells, but the twins do share immune cells. So the last time any twin shared anything with its other twin was before birth. And before birth, we also know that the enzyme that makes insertions doesn't work, or at least doesn't work that well. It's essentially turned off. So then we looked at the number of insertions in the most abundant clonotypes, the ones that have the biggest counts. So if you remember my power law, the ones that were on top of that curve. And we looked at the number of zero insertions in the most abundant clonotypes as a function of age of individuals. We have this amazing individual who's 101. So, okay, and then we can basically see there's a decay of these zero insertions. And our assumption is that the zero insertions, some of them are produced later just by chance, but a big fraction of them come from your prenatal life, because in prenatal life, that's essentially most of what you were producing. And with that assumption, we find that the clone lifetime is of the order of 36 years. So not each cell, but the clone gets reproduced for 36 years. So basically, more or less 1/3 or half of your life, which is a very long time. This is definitely not what people were expecting. The other thing that is known to happen is that the naive repertoire decreases in size. So we did check for the fact that it's not just caused by the fact that the naive repertoire decreases, and that's the red curve. So this is a control, which basically proves the fact that thanks to twins, we learn that these cells are very long-lived, and your repertoire is produced and then hangs around for a long time. And just by picking a small subsample of a very large space and then propagating it for a very long time, we all end up being very different, although statistically the same. So I think with that, that's my message. Thank you.
I didn't get, you seem to make a difference sometimes between repertoire space or recognition space and sequence space. What exactly is the difference?
What's the difference? So, okay, recognition space is an abstract concept in which just a pathogen and a receptor, if they're close together in this space, they're going to recognize each other well. What we have from experiments is actually a sequence, which is a real entity. And then we don't know if I test a given sequence against a real antigen, I can't tell you whether they'll recognize each other well. I can have the sequence of the receptor and I can have the sequence of the pathogen. I can calculate a distance in that space, but that distance is it's completely uninformative of the functional distance. So the functional distance in biology is called the phenotype, and this distance, the sequence distance, is called the genotype. And the problem of mapping the genotype to phenotype is one of the biggest problems. The optimal part I did because I was doing everything theoretically. I did in the space that interests us, which is this phenotypic part. But the data I have in this, sequence part, and then we are actually doing an experiment that is trying to map the two together and see what the relation is, because that's the missing link.
At the beginning, you said that V and J are correlated. So does this play a role in controlling the distribution of the repertoire?
For the distribution
you said that V and J are correlated. Does this play a role in controlling the distribution of the repertoire?
So it's D and J that are correlated. It plays a role in the distribution just because certain combinations are forbidden. It limits the diversity. I mean, it's basically linked to the architecture. You have D1 and then 6 J templates, and then D2 and the next 6 J templates in the genome. So if you recombine them, if you pick V2, you only have access to the next 6, whereas if you have V1, you have access to all of them. So it does limit the diversity, because if you've picked D2, you don't have the rest.
Do you think it's just an important biophysics constraint, or is it useful to, precisely to limit this repertoire, which is still too high at the beginning?
That's an interesting idea. And so, as humans, we have less D genes than either mice or fish. Again, this thing that comes up in biology, just if you have more doesn't mean you're better. Not to say that we're better than fish, but more complex. So the D gene is also an interesting thing, because it's very short and often it gets completely deleted. So why is it there to begin with? So, your idea that maybe it's there to limit somehow impose a number of combinations, and just, in fact, that its precise form doesn't matter, but it sort of puts you down one branch of the chosen tree. That would be an interesting idea, yes.
You have described the size of the repertoire, but not the size of the pathogens.
Yeah.
Isn't that bigger?
Well, this is the whole problem, I think, in many areas of biology. What is the environment? In neuroscience, it's a problem. So a lot of the ideas, and this is sort of also what I had in mind, showing you that the problem of sensing is omnipresent in biology, because a lot of times we have the idea that it's the matching between the statistics of the environment and the statistics of the response. But we have no idea what are the statistics of the environment. Even if you think about your well, vision or olfaction? How do you try and quantify the diversity of natural images, of natural smells? And now, how do you quantify the diversity of pathogens? You know, even if you could fish all the pathogens in a room and sequence them, you know that's not everything because you're going to move, the season's going to change. How do you quantify that? So all of, you know, if we had this side of the problem, we would be very happy. But we're trying to learn about this side of the problem from sampling this side, so the immune part, the response part at different times. And what we're doing, in fact, now is we're trying to make some assumptions that if we see a given response now, we can assume some form of the environment, let it evolve, and can we make a prediction about the future form of the environment and the future form of the response?
But isn't it true that if the sizes of the repertoire and of the pathogen were very different, we would either die in infancy or...
No, so that's another,
forever?
So this is actually another fascinating direction of, you know, okay, we could die, that's not the biggest problem. But the other problem is why don't the pathogens die? Either why don't the pathogens die or either why don't we die? Why are we constantly engaged in this tug of war and why are we both still there? And this is also one of the problems that I'm interested in looking at, and I don't think any. Well, nobody has won for all of it. You know, again, let's not think about us because you know we're recent and and unimportant. But bacteria have pathogens too, and they've had many, many more billions of years to win or lose. So why haven't they? And so there is something setting the equilibrium. You could, you know, some people think, oh, because some species from the general pathogen family are not actually pathogens, but I say commensal bacteria that we need. You know, so maybe but nobody knows. You know, how do we keep these two in check? It's a fascinating question.
Just wanted to ask a last question, bringing back from this very general discussion to a very technical one. So when you have this model of recognition, so you have this matrix FRA, and so if I understood correctly, you give a kind of one-dimensional or two-dimensional structure to this matrix. So you assume that a given receptor, is this natural from some biological point of view?
No, so it's another, okay.
I mean,
why do we do anything in 1 or 2 dimensions? Because we physicists, and in 1 dimensions, we actually often can do integrals. And especially if we make everything be Gaussian, right?
Sure.
Then life becomes, the Gaussian world is very attractive. We didn't just limit ourselves to Gaussians, but if we make everything Gaussian, we can actually do everything analytically in 1 dimension. We started off with 1 dimensions, looked at 2, nothing changed. The question, so all of this is the dimension of this effective recognition space. And nobody knows what that dimension is. Perelson, about 20 or 30 years ago, had estimate. Well, he didn't have estimates. He suggested maybe it should be of the order of 5, but this was an argument based on looking at how many different biophysical directions he can come up with, and okay, it's basically a random guess. But if you think about it as a dimensionality problem, you know that in high-dimensional spaces, there's many things that are not a problem, but covering a high-dimensional space very well is essentially impossible, right? Avoiding things in high-dimensional spaces, there's also the problem that you do want to avoid yourself proteins, so avoidance is completely not a problem. If you're in too low a dimension, well, it's probably recognition space is probably not 1-dimensional. So you can ask the question of, you know, can you sort of develop this framework, and this is also something that, you know, we've been thinking about, and try and estimate whether there's a sort of optimum trade-off for the right dimension of recognition space. Then there's a question of how do you test this experimentally, because it's a very effective space. And again, that gets back to Giuseppe's question of this phenotype, genotype mapping. But these are, and if you want to talk about packing and things like that in in these dimensions, I'd be very happy to.
Okay, I think we can thank Aleksandra.